Okay, we're going to go ahead and get started. Good afternoon, everyone, and welcome to the NINDS Hill Pain Therapeutics Development Program webinar. My name is Wakanda Metley, and I am the Operations Coordinator. We will start off with a presentation of slides, followed by questions and answers. Please submit your questions in the chat section featured below, and they will be answered at the end of the presentation. The presentation will be recorded and placed on our YouTube pages in the coming days. I will be sending that link along with the presentation slides to everyone that has registered. Please remain muted for the duration of the presentation, as well as keeping your cameras off. I will now turn it over to our moderator for today, Dr. Marianne Palamonter.
Thanks, Wakanda. Good afternoon, everybody, and welcome to the webinar that's going to be describing the Heal Pain Therapeutics Development Program. The Heal Pain Therapeutics Development Program is part of the Helping to End Addiction Long-Term Initiative. The approach that Initiative uses to helping end addiction long-term is to, number one, enhance pain management, and number two, to improve prevention and treatment of opioid misuse and addiction. I like to focus, of course, on the enhancing pain management aspect of this. And the four major research strategies HEAL uses to enhance pain management are to understand the biological underpinnings of chronic pain, to accelerate the discovery and preclinical development of non-addictive pain treatments, and that's what we're going to be focusing on today, and three, to advance new non-addictive pain treatments through the clinical pipeline, and then four, to inform best practices for effective pain management while minimizing the risk of addiction. So if we're just talking now about the analgesic development programs, the HEAL non-addictive analgesic development programs cover pretty much the entire early drug development spectrum, from discovery through IND enabling and finally into phase 1 and phase 2 clinical trials. I want to highlight some of the funding opportunities that are in the earlier stages, and then also one that's in the later stage. So there's an R61 planning grant that is focused on the very early stages of discovery, and it covers things like planning for future grants on the same topic, looking at the preliminary biology and mechanism of action, and just assembling all of that data. Then there's also a U19 team research grant that covers that aspect that the planning grant does, and actually the planning grant is supposed to feed into this U19 grant. And this U19 covers assay development, screening, initial hit characterization, and biomarker development. And then what we're going to talk mostly about today, which is the Pain Therapeutics Development Program, that covers, again, some early aspects of discovery from hit to lead and small molecule language, all the way through a phase 1 clinical trial. And then there's some additional programs that support the development of translational assays like organ on a chip, human cell cultures that have to do with pain, like DRG cultures. And then and that's through NCATS. And then there's another program called the Pain Preclinical Screening Program that covers in vivo preclinical therapeutic profiling for pain. And then finally, there is a phase 2 opportunity that I'm not going to talk about today called EPPIC-Net. There are 10 participating NIH institutes, NINDS, NIAAA, NIDA, NCCIH, NIAMS, NICHD, NIDCR, NIDDK, NEI, and NHLBI. So just to give a little more detail on the R61 planning grant and the U19 team research grant, that planning grant supports up to direct costs of up to $500,000 per year. It's only a 2-year duration because it is for planning and getting your team together. And so some of the goals for that are to build a multidisciplinary team that could be utilized in the team research grant, and then also to obtain preliminary data that addresses the entry criteria for the team research grant or for other grants. The team research grant, that's the U19, entry criteria for that are evidence that you've built a multidisciplinary team and that you have the expertise to do that and to work together. And then also to have a strong biological rationale that addresses the feasibility of what you want to do. And that should be evidence for that should be good preliminary data and or literature-based evidence. The team research grant is a 5-year maximal duration if milestones are met each year. And it supports up to direct costs of 1.5 million per year. And renewals and revisions are not allowed for either of these grants. If you're interested in these grants, which is, I should stress again, are the earlier stages for drug discovery, you should contact Julia Bachman, and her contact information is on this slide. Then I want to say a little bit about the preclinical screening program for pain. This provides a platform to identify and profile non-addictive, non-opioid therapeutics for pain. It takes assets that are biologics, small molecules, natural products, or devices, and profiles these assets in a very rigorous way, allowing them perhaps to move towards clinical trials once the profiling is complete. And the way it works is under NINDS direction, rigorous preclinical evaluation of assets are conducted in a suite of assays performed by a contractor, Psychogenics, which a lot of you are probably familiar with. This testing is performed in a blinded and confidential basis, and it's at no cost to the participant. The PSPP staff interacts with the participant to plan testing, review the test results, and discuss future studies. So the PSPP staff has quite a bit of interaction capability on this particular program. And then finally, an important thing is that the intellectual property of the participant and confidentiality are protected. PSPP is open to academic groups, industry, and government asset owners worldwide. And if you're interested in this, you should contact the program director, who is Smriti Iyengar, and her contact information is on this slide. Then finally, to start talking about the Heal Pain Therapeutics Development Program, or PTDP, one important thing to notice right away is that the support from this grant covers a large swath of discovery and early development. So it covers everything from hit to lead optimization all the way through a phase 1 clinical trial. It uses a virtual pharma approach that features NIH industry experienced consultants and NIH contract resources. It supports biologic and small molecule therapeutic development, but it does not support device development. NIH consultants are assigned and tailored to each project that's funded based on the needed expertise for the project at whatever stage it comes in. And the NIH contract resources are also tailored to whatever stage the project is in. If you are awarded one of these grants, you also have the choice. So you can choose which NIH contracts to use, or you can opt out of using any of those contracts and budget in your own contracting in the grant proposal. It's also important to mention here that your team's intellectual property is retained by your institution. We do everything we can to make sure that occurs. Then I also wanted to lay out what happens if you get awarded one of these grants. So the grant, everything revolves around the grant itself. And once you're awarded one of these, a lead development team is formed around you and your team. So people who are on this team are you and your team, also the NIH industry seasoned consultants who have expertise ranging from assay development to med chem to CMC to pharmacokinetics and to finally pharmaceutical development. You also are assigned an NIH staff project manager and subject matter experts. So this diagram just kind of shows you the variety of CROs that are involved with our contracts. So we have contracts that are focused on medicinal chemistry, and that our vendor is Curia. PK Tox, we have 3, actually we have 4 vendors now. Data management, CDD is our vendor for manufacturing and formulation. We now have multiple vendors, and the reasons we have so many vendors for the manufacturing and formulation PK Tox is that we've just added vendors that do biologics as well. So now we're set up pretty much completely for biologics and small molecule therapeutic development. And then we have 2 vendors for phase 1 trials, that's Duke University and PPD. Then I just want to say a little bit about the grant that all of this revolves around. The funding opportunity is called RFA NS 21-010. And the title of it is Non-addictive Analgesic Therapeutics Developments for Small Molecules and Biologics to Treat Pain. And it's called the UG3 UH3, and I'll talk about what those 2 things are as we move forward here. The goal is to accelerate development of novel non-opioid, non-addictive analgesics. And some of the features of the grant include that it's a phased grant, and that's where the UG3 UH3 part comes in. The UG3 is a shorter, more early phase that enables things like just getting everything together. It's more preparatory. And the UH3 is the execution phase. And I'll describe those a little more later. And it is milestone driven, this grant, and we'll talk about that more. And it's also what's called a cooperative agreement grant, which means that, again, NIH staff have quite a bit of input into how things and how the program works. The grant supports a variety of activities from hit to lead, lead optimization, lead selection and characterization, also supports biomarker optimization and PK/PD development. Finally, also supports IND enabling studies and phase 1 trials. The end goals of this grant and the program is to identify and fully characterize a lead, identify target engagement biomarkers and use them, seek partnerships because we want you to get past phase 1. We want to make sure that everything that we can do to facilitate getting you approval from the FDA is one of our goals. So another goal is to complete IND enabling studies, file an IND, complete the phase 1 trials, and have you ready for a phase 2 clinical trial. So another important thing to remember about this grant is that applications can enter in either the discovery or the development stage. And when we're talking about the discovery stage, we're talking about exploratory hit to lead and lead optimization. And when we say development, we're talking about IND enabling and phase 1. All grants, whether you enter in discovery or development, must start with the preparatory UG3 phase. And we'll talk more in the next slide about what you actually do in that UG3 phase. And because it is, of course, different depending on whether you enter in discovery or development. And the combined duration of the UG3 and UH3 phases is 5 years maximum. Usually, people apply for either a 1 or 2 year duration UG3 phase. 2 years is the max. And then whatever is remaining of that is for the UH3 phase. So most people have a 2-year UG3 phase and a 3-year UH3 phase. And it's important to point out that when you write your application, you must outline your approach and your plans for both phases, and milestones for both phases have to be in the application. Okay, here are some of the things about what makes you an eligible organization. So academic institutions are eligible, small businesses are eligible, and foreign institutions and foreign components are also eligible. But we'll say here a little bit more about foreign organizations. So there is more scrutiny if you are proposing to use a foreign institution as the primary receiver of the grant or as a component. There's the most scrutiny is if you're trying to have a foreign institution as the primary component. That is, I'll just say it's quite difficult. You have to have a very, very good rationale for that. If you want to have a foreign institution as a component, it's just important to point out what's unique about that institution, why it's an important part of your application. Sometimes things like where a patent is makes it important, and sometimes just unique resources, but you have to make sure that's clear in your application. Then here are the entry criteria for the discovery stage or the development stage. So remember, you could enter in one of these stages. So if you enter in the discovery stage, you need to show that you have a rigorous biological rationale for the intended approach. You need to show that you have a promising small molecule or biologic starting point for optimization, and that you have scientifically sound assays to optimize and test the agent. If you come in at the development phase, then it must be very clear that your candidate therapeutic is identified, and there should be a strong data package in your application linking modulation of the therapeutic target by the candidate therapeutic to disease modification. Biologic activity and admet properties of the candidate therapeutic also have to be shown that they're appropriate for the intended use. And you should be able to, in your application, give us a table that shows all the information on your candidate therapeutic if you're coming in at the development stage. And then some applications, there are some things that are not responsive to this funding opportunity, which means that they will be withdrawn. The application would be withdrawn. So if you're screening for initial hit identification, the U19 actually covers that, and there are other grant mechanisms that we offer that cover that. So that's not responsive. If you want to do basic research and studies of disease mechanism, also not responsive. Same way with animal model development, development of diagnostics and diagnostic devices. Clinical studies that go beyond phase 1 testing, we do not support because we have EPPIC-Net. And then if you're proposing an opioid sparing project or projects that target the mu opioid receptor, that is also not responsive. So important to keep that in mind. So here's some typical activities in the UG3 phase. One is optimization using potency and efficacy screens. Another is showing preliminary efficacy and doing tox testing in appropriate animal models for pain. Characterizing and testing for in vitro ADME, and ADME of course stands for absorption, distribution, metabolism, and excretion. Conducting initial optimization of a pharmacodynamic or target engagement biomarker that's associated with your therapeutic target or pathway. In the UH3 phase, when you've done all of these things, and again, a lot of this depends on whether you're entering in discovery or development phases. But in the execution stage, any further optimization activities as listed above can be completed. Pharmacokinetics, biodistribution, gene expression, tumorigenicity, and immunogenicity can be tested there. Cell bank development and testing is part of the execution phase, as is formulation manufacturing for toxicology and human clinical studies. And then, of course, IND enabling characterization of the clinical candidate, non-GLP and GLP tox, and IND submission and phase 1 clinical testing are all activities typical of what you do in the UH3 phase. And you can see there's a long list here in the UH3 phase. So if you come in the discovery phase, I mean, it's typical. That's why a lot of people allow themselves 3 years for the UH3 phase and 2 for the UG3 phase. Again, if they're coming in discovery. So then another important thing to understand about this funding mechanism is that we mentioned that it was milestone driven. And so in order to advance from the UG3 phase to the UH3 phase, there is an administrative review. So you don't have to go through, you don't have to submit another application like that's similar to what you had in the initial one. But you do have to submit what's called a type 4 application, which just goes through administrative review. It does not go through peer review. And so part of that is that the NIH program staff and leadership will be reviewing your UH3 application, or just how well you've achieved your milestones based on, again, have you met them or have you not? looking at the overall feasibility of project advancement, including biomarker work, and considering data that might not have been captured in milestones. They'll also be based on competitive landscape for the disease indication and drug target, program priorities, and availability of funds. Because, keeping in mind that HEAL is a very broad program involving many agencies and institutes.
Done for next year.
Okay, could people please turn off their audio for now? Now, just to talk about the milestones, what do we mean when we're talking about milestones? Again, remember they're an important component of the administrative decision to transition to UH3. And also keep in mind, we do evaluate whether people are meeting their milestones on a yearly basis as well. Milestones, therefore, should be included in the application, and they're used for measuring success in an objective way. So there should be one or more milestone that is used for each key objective in your approach section. For each milestone, it's very helpful to provide details on methods, assumptions, experimental designs, and data analysis plans. It's also important to specify something that's related to a quantitative criteria for measuring success and related rationale. A good example is if you're talking about, a screening funnel and you're trying to explain how you make a decision about which compounds or which biologics proceed to the next stage. What are the threshold cutoffs? Maybe it's affinity. So what's the affinity range that you're willing to accept to move forward? So that's just one example. So these quantitative criteria should be robust and consistent with the state of the art in the field. The quantitative criteria for success and milestones will be used for making go, no-go decisions, and this should be specified. You should also specify the timeline for each milestone. Again, one milestone per year. And then another thing to remember is even though you're putting these milestones in the application and the review panel, the peer review that looks at your application will look at these milestones as just part of an overall evaluation of your application. They will be further refined after the award is made with the lead development team that has been formed around you and NIH staff. Here a little bit about intellectual property expectations. Inventorship is determined per US patent law. NIH Pain Therapeutics Development Program itself has no stake at all in the intellectual property. Prior to grant award, your institution must have upfront IP agreements in place with all potential inventors. And these agreements must address who will hold title to the intellectual property on new chemical matter in use or new biologics, and what royalty arrangements will look like. Also, the intellectual property agreement should aim for unencumbered IP consistent with our program's goal to create a licensable product. So now please ensure that your application has separate budgets for each year of the grant. So if you have a grant that you can propose a grant, I should mention, that's only 4 years. You don't have to go the full 5 years, but let's assume for example's sake that you choose a 5-year grant. So then there should be 5 separate sets of budget sheets plus the cumulative budget in your application. You should also clearly indicate within a table which activities will be conducted by you and associated personnel on your team, and also which activities would you want to be conducted by the NIH CROs and or consultants. You should also have a table of proposed activities that provide information such as activity, optimization effort, assays, PK, et cetera, throughput, sources of activities like who's going to be doing what, and advancement criteria. So all of this should be clearly laid out. Not everybody uses a table, but it really does help the reviewers. You should also include, everybody should include a target product profile table, which talks about the clinical characteristics that are acceptable and ideal that you are shooting for, at least at the beginning, with the understanding that target product profiles will change over time as you gather data. And your application should also address a biomarker component. It should address intellectual property, and that should be in the other attachments section, not in your research strategy section. And then if you are asking for NIH to conduct a clinical trial under our contracts, our clinical contracts, then mark application clinical trial as no. If you're going to do your own clinical trial and you've budgeted that into your application, then most often you would indicate that it will be a delayed onset clinical trial. This is just something to remind you, when this funding opportunity was initially put out, actually when the earlier version was put out, there was a cost matching requirement for small businesses through HEAL, and that's now been removed, so you don't have to worry about that anymore. And so pointers, things to do, read the funding opportunity announcement carefully, really, really look at that. And then even if you've read it carefully, please talk to us. So our contact information is in the funding opportunity announcement, and it's also in these slides. So do not hesitate to discuss your proposal with us. We will happily set up a call with you. Stick to the page limit for the research strategy. I think it's 12 pages. In your scientific premise, explicitly discuss the quality of the data presented in prior publications in a detailed manner. And this kind of leads to the whole rigor idea here, which is really important to all of NIH. Make sure you detail the controls being used for each type of experiment and appropriately highlight potential confounds like surgical exposure, genotype, culture to culture variability, and human placebo effects. Include details within the experimental design about the reduction of potential bias, including blinding, randomization, and what your inclusion exclusion criteria are. And then finally, describe the source of the data on which the sample size estimation or power analysis was based, and provide some details about the power analysis itself. Don't just say, we're going to do a power analysis. Give a little more detail than that. And then some additional things to do are to complete your required registrations at least 6 to 8 weeks in advance of the receipt dates. Consider submitting your application early. This gives you a chance to react to issues that might result in your application being withdrawn, just in case a mistake's made or something happens. Discuss your budget with NIH staff if you have questions. Definitely talk with your tech transfer and business development group, so they can help you a lot with your intellectual property plans and your data sharing policy. And then you need to plan for funding patents and licensing activities. Things not to do is, definitely do not plan for this to be the sole funding for your lab, because as we mentioned, it is a milestone-driven program, and if you don't meet your milestones, the program can end abruptly. And also, don't forget to make use of NIH contract resources, and make sure you understand, if you need to, that is, and also make sure you understand what we offer too. Don't plan for NIH contracts that solely look at disease biology. Usually that is what the application of the grantee is supposed to provide, although with our new biologics contracts, we do have some disease biology that are part of them. And of course, there is the PSPP as well. Please do not include NIH contracts in the application budget. So if you say in a table that you want to use our contracts, all you have to say is that. You don't have to put like a design or anything like that, because that's what we will do with you in the LDT. And then do not reach out to planned NIH contractors for letters of support. We are the people who are the sponsors for those contracts, so you don't have to worry about that either if you're using our contracts. If you're using your own contractors, you should provide letters of support and you should detail your designs. All right, so that's it. And so for contact information, this slide has all of it on. Again, I'm the program lead, and I'm the person who you should reach out to when you have questions about these grants. Chuck Sywin is the director of the Small Molecule Neurotherapeutic Development Program, and the HEAL program is under that program. Carolyn Bonder, who's on this presentation right now, is our health program specialist. That's her contact information, and Rakonda Medley is our operations coordinator, and that is her contact information. So now please feel free to put questions in the Q&A, and we can start that part. So do we have any questions yet, Rakonda?
Nope.
Yeah.
Yeah, no.
Not yet. Okay.
Also, for those that joined in later, the slides will be available to you all sometime next week.
I can ask a question to get it going, Marianne. Are there any budget limits for the UG3, UH3 phases?
Right, that's a good one to cover too. And so there are no budget caps with either of those phases, but the budgets must be very well justified. Looks like a number are coming up now.
You mentioned we need to find partners beyond phase 1. Can you elaborate?
So since we want you to make sure that if you have something that's gone as far as phase 1, it's got a lot of promise. So we want to make sure that this gets to patients. So it's good to work with your tech transfer office. That's typically how people start this, to find out if you can partner with anybody, if there's a licensing opportunity available. Give yourself some training to how to pitch your project to potential partners. And then of course, keep in mind that the EPPIC-Net program could provide phase 2 support as well. So I hopefully that makes it clear.
What is the appropriate career level for the PI of this mechanism?
So, I mean, technically, you need to have an institution supporting you. Most people who apply are at least at the assistant or associate professor level, or they're in leadership levels in a small business. Because one thing that gets evaluated by review is the ability of the team from the application to actually conduct the work. And so you have to have people that are experienced enough and qualified on the team to be able to do the work. And that is a review criteria.
How is the actual NIH division determined for assigning an application?
So in this particular case, because it's HEAL funded, it all goes to the same place. So it all goes to NINDS. And I mentioned earlier that all of these other institutes had signed on. So for instance, if you want to do something that has to do with osteoarthritis as a pain indication, NIAMS, that institute has already signed on to this. So what we would do is we would add a program officer from NIAMS to the project team. So I would always be the primary program officer, and then we would add a secondary officer representing the appropriate institute. Same way with NEI, for instance. So, but they're all signed on to this grant. So it's easier for you this way.
And Peter asked about the recording, and that will be provided to you by Rakonda after this session. Okay, next question. Yearly milestones may not align well with milestones associated with project components. How is that dealt with?
So what we do, yearly milestones, we have to have some way to get you started thinking about what you need from year to year. So that's why we do yearly milestones, but they can be altered as you get data in. So, I mean, they can't be altered right before you're going to be evaluated, of course, but if right when you start your lead development team, you will, get a lot of input from the consultants that have been assigned to your team. And you will also be presenting an introductory presentation to an external oversight committee that we have that consists of experts in pain therapeutics development who were in very senior levels in the pharmaceutical company. and pharmaceutical industry. So they will also have some suggestions, and that's why it's important to have your TPP ready to go. But you'll find that yearly milestones, we're flexible depending on the data, but you'll find that they help you plan.
Would the program be suitable for the development of an evidence-based medical food that stops the transition from acute to chronic pain?
So usually we take in therapeutics that would go through FDA regulatory pathways. And so I'm not sure, I mean, if it was a natural product, yes. But, then it would be still subject to toxicology studies, IND submission, and so on. If it's a food that doesn't go through those regulatory pathways, it doesn't really fit what we can offer. So I'd say that maybe not.
Are there sample applications available?
No, there are not actually, unfortunately.
You could potentially, we do have our awardees on the site, but we can't provide their.
No, I mean, you can get abstracts off NIH Reporter, but I think you might understand, since we really want to make sure that we protect the intellectual property for you as the applicant, we just have to be very careful about confidential information.
Please elaborate on the review process and timelines.
Okay, so the review process is everything goes to the same so once you submit an application, everything is routed to the same review section, and it's a SEP, a special emphasis panel, through NINDS. And that panel consists of people who have expertise in pretty much all the cross-functional areas you'd find in a pharmaceutical company, and also emphasis in the pain area that your application's in. So each time a round of application comes in, the scientific review officer tailors the review panel to make sure that whatever, your therapeutic modality, let's say you've got a delivery method that's unusual, or you've got a pain condition that is very specific, we make sure we get expertise that covers all of those things. And then that is why when you get your review comments back in your summary statement, you may have 5 reviewers that have comments and have scored you, because we're covering all those different areas, drug discovery plus the pain condition area. And we do not use percentage scores, because these are such, usually they're much smaller than what goes through CSR. So we just provide the impact scores.
And that's the next question. What score is in the fundable range?
So that varies from cycle to cycle. So it's kind of like being graded on the curve, I guess you might say. If you have, I haven't seen any in ours that are awarded if they have scores of 5 or above, but that could change too, depending on what the program's needs are and what funding looks like.
Is this a good program for projects that have begun phase 1 clinical trial but need additional funding or assistance?
I would say no, because if you need additional funding to, like, let's say you need to bolster your efficacy profile because you only maybe used one pain model, then the PSPP program is a perfect place to go, because that can provide you the rest of that data. If you need reproductive toxicology or something like that, unfortunately, we don't offer that. And that you probably have to find elsewhere.
Please provide any additional information about the R61 if we have a lead compound that may have an analgesic effect. How much preliminary data do we need to apply for the R61?
So I first want to see if Julia Bachman is on this. Is Jules on?
I don't believe so.
Yeah, I don't think so. So my understanding of that is that it is always helpful to have some preliminary data, but this is the very earliest in our set of funding opportunities. Other than, I guess we do have an R01 where you could, I guess, validate your target, but in general, you don't have to have a ton of preliminary data, but I think it's just by nature, having some and making sure it's rigorous is always helpful.
Okay, one more. How many compounds can I propose for optimization or development?
So if you're still in the lead optimization stage, to us, we're still doing analoging, we're still doing some SAR, so that you can do what you want. You just have to have a plan that makes sense. If you're in development, though, we can only handle one clinical lead that we take forward for IND enabling studies.
I think you already answered this, but do we need prior authorization for direct costs of over 500,000 per year?
For this particular grant, no, you don't, because it's a set aside with HEAL, so you don't, and it's an RFA, so you don't need that.
Okay, I think we'll give it another minute. I put the R61 FOA link in the chat, so if you're interested in reading more about that, please visit the site with the FOA.
And then I would suggest too, if you're interested in that, you should contact Dr. Bachman. I thought there was another one, Carolyn, that came in.
Just right now, yep. Just for clarification, if we are in IND enabling studies with a candidate, can we do some SAR for backup?
So if you're being funded in development because you're in IND enabling, the answer is no, we don't support that.
I think that's it, Marianne.
Okay, I guess we could give maybe 2 more minutes or so to see if there are any more. And again, just to emphasize, the best thing to do if you're interested in any of these grants is to contact the program officer that's listed in these slides.
Okay, if I'm not using contractors and don't need consultants, do I still need to have a lead development team?
Yes, that's one of the hallmarks of our program, because even if you're not using our CROs, you would still be assigned a consultant, and you would still, these lead development teams meet twice a month, and we really feel that that helps guide you in a relatively short time towards the goal of an IND submission. Well, I guess,
One more.
oh, here's more, one more. Okay.
Does the program allow for multiple PIs?
Yes, absolutely.
And I'll just remind everyone, the next application due date for this grant is February 14th, 2023.
Okay, if there are no more questions, I'd just like to thank everybody for your interest and thanks for listening to this presentation. Good luck with any of your applications you put in.
Thank you, everyone. Have a great weekend.
Well, have a good weekend, Carolyn.
You too. I think you can end the meeting.
I'm gonna, yeah.
Okay.